Hello, my name is Miguel Castresana Aguirre and I am a PhD student working for Stockholm University in Lab Institute. I will be presenting a poster with the name Funcoup 4.1 Networks of Functional Gene Associations. In case someone doesn't know what Funcoup is, it is one of the most comprehensive databases for genome-wide functional association networks. Funcoup is crucial because it identifies functional associations which gives important insights for the understanding of higher level mechanisms performed by complex cellular processes. Funcoup offers associations for 17 different organisms across 10 different evidence types. For the workflow of Funcoup, networks are inferred using 5 different known interaction classes, such as metabolic and signaling pathways extracted from KEGG database, protein-protein interaction, and shared protein complexes and shared operons. Associations are inferred for 10 different evidence types using a redundancy-weighted Bayesian integration approach. Important to mention that Funcoup uses orthology transfer between the different species. The final inferred network gives associations between genes, and these links have a probabilistic confidence score ranging from 0, no association, to 1, a high likelihood for an association. As you can observe in the right part of the slide, we have the species that Funcoup has, and on the x-axis we have the contribution to the final network for each of the evidences that Funcoup provides. Very important to mention that, as different for other databases, Funcoup does not use text mining to infer associations, since it is known to be prone for false positives. As I mentioned before, Funcoup uses orthology contribution using a comprehensive database called InParanoid. This step is also very important because it increases the coverage of species that are not that well studied. Now, to demonstrate how powerful Funcoup is, I will end up the presentation with an iterative example. For that, we will go to our web server. Here, one can input the list of genes of interest. In my case, I will input human genes that are known to be part of a evolutionarily conserved protein complex. One can also do an advanced search with many different properties. In my case, I will select a comparative interactomic approach because I am interested in searching conserved subnetworks in different species. In my case, I will select Mus musculus and Bacillus subtilis species. Here, once the search is done, we have different subnetworks for the different species that we selected. In my case, in blue, Homo sapiens, in orange, Mus musculus, and in red, Bacillus subtilis. We can see that the subnetworks from mouse and human are very well conserved. Almost all the genes that are in the human subnetwork are found in the mouse subnetwork as orthologs. We can also observe that the complex that I input is also found in mouse subnetwork with their corresponding links. However, in the Bacillus subtilis subnetwork, we find that almost no orthologs are found, only 3 of them, and we have that only half of the complex is found, having those genes no links between them. This example is powerful because this shows the utility of Funcoup, because this lack of network conservation in prokaryotes, in this case Bacillus subtilis, suggests that this complex arose in the eukaryotic lineage. Then, apart from the nice network visualization, Funcoup provides much more information in the following windows that you can observe. In case someone wonders from what type of information my association between two genes come from. One can go, for instance, I want to know the interaction between LAC-TB2 and hint, from what evidence type my link is coming from. We can observe that the highest score is obtained for mRNA co-expression. Then one can click in the following box, and it will even give you from which experiments this score is retrieved. Finally, we can also have enrichment terms, for instance for KEGG signaling and metabolic pathways, and also for geo terms. And that's all for this presentation. Hope to see you enjoying our functionalities soon.